Welcome you all to this cryo-EM course. Last class we discussed about the lipid protein aggregation, high concentration of the protein sample, as well as I showed you some good negative stain images, and now we want to discuss more about the application of this negative staining method, how we can use this negative stain for our research purposes. So, this is one of the sample I was showing you in last class also, that this is one of the sample. This author mentioned that this sample is forming a tetrameric assembly. So, last class also I selected this particular image and I showed you how tetrameric assemblies are there. Here you can see there are many small particles are there. You have to always consider that you can say that there are some different shapes are there. So, here different orientation is one of the important factor. When you have a carbon layer, and you will always get a same orientation. If you look at my hand, this example, it will clear to you. So, you just consider this is a protein sample, this particular thing, and now it is a rectangular shape. That rectangular shape we are discussing in our TEM images. Now, in a carbon layer, we are always expecting that your protein will stay like that. But what about this? When protein will stay like that, you will see only this particular view, and that view basically thinner than this particular view. Or if your protein is just attached to the carbon layer like this one, you see only one part of that. You cannot see all the very thick flat like that. So, exactly same thing is happening here also. If you pay attention to those, here you can see there is some rectangular shape, here. So, some rectangular shape proteins are here, but same time there are some small proteins are here. So, that is the different orientation of that protein. So, if we look at this or this one, basically it is showing you the same rectangular shape of the protein, and you can see one side of this protein, what I was showing using my phone. This is the rectangular shape of your protein, and from this one author just mark different domain area, which is helping us to which is helping them to understand how many protomers are interacting together. Here from this different color you can tell that okay, there are total 4 protomers are coming. So, here one protomer, here the another one protomer is interacting, here one protomer, and the 4th protomer are connected. So, from this negative stain we can tell that okay, we can see a rectangular shape protein where 4 protomers are interacted together and forming this large complex, as well as if you just rotate this protein from it is like this form, it is there. Now, you are rotating 90 degree here. So, you can see the other view, which other view you can see those molecules. So, this is the another view of this same protein. So, from this one you can get an idea what is the orientation of these protein molecules as well as how they are interacting together. Now, next we will discuss little bit more about one more protein sample. So, if you have a negative staining images like that, from there you can do some this kind of analysis where you looking at this individual protein molecules and you analyze those data and you calculated the 2D averages. From there you can tell what is the oligomeric state of that protein, and here the oligomeric state you can visualize that. So, from that ACC profile you can calculate the oligomeric state. From the sigmoles also you can calculate the oligomeric state of the of that protein, but here you can visualize what is the actual oligomeric state of that protein. Now, in the next one, that here as we mentioned previously that okay, mostly we perform the ACC purification. This ACC purification basically give us give us that idea that okay, this is the area from where we want to collect the sample. And then this is the antigen antibody reaction. If you just consider this is a HIV trimer, so this HIV trimer with Fab fragment we incubated, and HIV trimer means we have this kind of trimer and it is interacting with the Fab, and that we are isolating here, that okay, when we have the only HIV trimer we perform this reaction and then we perform the ACC purification. From there we isolated this HIV trimer, and when we are running in SDS page gel we can see that two bands, one for the trimer and one for the antibody also. And here there is a binding study. Those are not important for this particular course, but definitely those are the biochemical study we should always do before taking that protein for TEM imaging or cryoimaging. Now here you can see individual HIV trimer. There are many individual HIV trimers are there. Now in this individual trimer, if you just look at here, this trimeric view is very clear. Now if this is a trimer, when we added the antibody, you can see overall size of this antibody, overall size is much bigger. If you just consider the trimer shape is only this much, but when you added the antibody, overall shape is much bigger, where you can see the overall shape of that molecule when you incubated this HIV trimer with the two different antibody. This is the enlarged view of these things, where this is the part of HIV trimer where antibody is attached in this particular place. The same thing here also, this is basically our trimer and those are the antibody attached with my sample. So from this study what we can visualize, actually we can visualize that okay, so this is our HIV trimer. When we incubated with HIV trimer with the antibody, antibody is interacting, can we visualize this? Where this antibody is interacting, what is the overall morphology of this protein? We can determine all of this thing from this negative staining study. So negative staining study itself, it's a very good technique from where we can get this different kind of morphological difference between that two protein, that one is antigen and another one is the antibody. And also we can compare if there is a two different types of antibody we are using, where they are interacting, is there any morphological changes are there or not. Not only that, we can use this same technique to generate a very low resolution three-dimensional structure, where in this particular study we are showing that three-dimensional structure where this is the trimeric shape of that HIV trimer and where these are the area of the antibody. So this is basically trimer and those are basically antibodies are interacting. So but most of the cases overall resolution is very poor and we'll just get a very low resolution architecture and overall morphology of that protein complex. From this study we cannot get more than that. Here again I already showed this particular image as a good image. So this is the same spike protein we are talking about. So if you look at, this is a very nice negative stain image and well distributed particle, staining is very nice and individual protein molecules are very clear. Now if you look at this 2D averages from this one, that is I was talking about, that you can see a cone shaped molecule. Most of the cases you can see a cone shaped molecule. From this negative stain, sometime you can see that RBD, that receptor binding domain, open and close form also, if you perform those experiments properly. So now this is basically overall shape of the spike protein, and not only that, if you just go through you can see some antibody density also clearly. In many cases from these 2D averages you can see antibodies are binding at the RBD region. So this is the transmembrane region where it will bind to the transmembrane, and here this is the RBD region where the so the 3 RBD region is here where the antibody is binding. So from this negative staining TEM and this 2D classification, not only you can get the concentration of the sample or overall distribution, morphology, you can see the protein-protein interaction also. Here there is another example with a different types of antibody with the same spike protein, where you can see there are lots of individual spike protein with antibody. When you did this classification, you can see overall similar kind of shape, but then you can see 3 extra densities. Some cases you can see only 1 extra density. So from there you can tell that okay, there are some spike protein where 3 antibody is binding. Some cases it is only 2 antibodies are binding. Some cases only 1 antibodies are binding. So from this 2D averages you can predict those interaction, that okay, how many antibodies are interacting or how they are interacting, what is the overall morphology. From these are the enlarged view from those 2D averages, where we are showing the same thing, that we are showing the overall morphology of that spike protein and then the density of the antibody here. But in this particular image if you pay attention, there are some places, there are some small densities or free densities are there, some free densities here and there. So those free densities, basically free antibody or Fab densities are there which didn't interact with that spike protein. So if you just compare this particular image and the previous spike protein with antibody images, definitely previous spike protein with antibody images are much more homogeneous than this particular sample. So we can also conclude this kind of we can also get this kind of information from this negative staining images. Okay, not only that, if you there is another protein called Vibrio cholerae hemolysin. So here you can see there is a lipid density. This is the lipid density, and generally Vibrio cholerae hemolysin, it oligomerize in presence of lipid density, and you can see those are isolated protein molecules and those are the circular protein molecules. And if you come here and calculate 2D averages, in this particular 2D averages you can tell that it is forming a heptameric shape. So if you count, this is coming a heptamer, and all these classes if you calculate properly, most of the cases you will get it is forming an heptamer. But the same, there is another truncated version of this particular protein. When we calculated this one, you can see overall it is a spherical structure, that heptameric portion is disappeared. So this particular protein has a heptamer, and from this negative staining images you can calculate that it is forming one protein is forming a heptamer, other one is not forming a heptamer, it is forming some globular shape. And as well as you can see there is a pore at the center, that pore is available in both the protein samples. Now this is another example where you can see this is a lipid membrane. Again this is the lipid membrane which we are showing it here, the enlarged view. In this enlarged view you can see this liposome. If this is a liposome, this liposome is decorated with this small protein. This protein is interacting with the lipid membrane, and from these stem images you can identify how many proteins are binding and how they are binding, and they are actually binding with the lipid membrane or not. Not only that, in this large view also if you look at here, you can see there are many protein molecules binding to the lipid membrane, and there are no free protein. Most of the time if you add this protein, some protein are going and only attached with the membrane. So this kind of study definitely is very helpful if you do it properly. If you have a proper negative staining images, you can conclude many things, that okay, how this protein is interacting with the lipid membrane, how it is binding to the lipid membrane. If lipid is there and if you are incubating with the protein, can you see any free protein molecules or not? Most of the time you will get those kind of information from these negative staining images, and which will be negative staining images you can use comparatively higher dose rate also, and that is the reason, and you are using a staining solution, and that is the reason your contrast of the images will be much better, and that will be much clear if we do this kind of study using the negative staining technique. Here you can see one more example is a very nice lipid vesicle, you can see it here, and if you look at this sample here, there are so many protein molecules are embedded in this lipid. So you can see there are many protein molecules, they are clustered on top of this lipid. So this is looking a very nice image where you can see how these proteins are interacting with the lipid membrane, and when these proteins are interacting with the lipid membrane, the background you will not get any free protein. So from this condition you can also tell that what protein concentration is required which will only interacting with the lipid molecule, and there will be no free protein will be available when they are interacting, where they are interacting actually, and you can change the lipid composition also, and from there you can tell what kind of lipid composition is important for this kind of interaction. So there are few more examples, similar type of example here. You can see it that there is a different types of lipid membrane, and this lipid membrane is saturated with these different types of protein clustering. So here also you can see proteins are clustered on top of that lipid membrane. So there are few similar kind of examples are there. So if you just look at there is a other image where also the similar kind of distribution of that protein you can see all over the image, how they are interacting with the lipid. So in this particular case you can see at the edge of the lipid some proteins are interacting, but that many of these proteins are staying outside of this lipid membrane. So in this background you can see the full of this toxin molecules. You can see there is a lot of big patches where the toxin molecules, but the inside very few toxins molecules are there. So in this particular experiment you can tell that this particular construct of this protein, it is not interacting with the lipid molecule too much, mostly they are staying outside of that membrane. So here I am showing you some liposome sample preparation. We can prepare that liposome, and here you can see those are the control liposome. So one we made with a 10 PC, that is phosphatidylcholine, 12 phosphatidylcholine, 14 phosphatidylcholine, then egg PC and cholesterol. So here there is no other samples are there, so only that lipid vesicle. If you look at, those are called the lipid vesicle, free lipid vesicle. Here also you can see these lipid vesicles, and those are the circular shape, it has a definite shape, and you can make a exactly same shape that 100 nanometer or 200 nanometer or 300 nanometer. Nowadays preparing that lipid vesicle is very important, because many cases we are using this as a drug delivery system. And now if you incubate that same lipid molecule, if there is a lipid molecule here and you incubate it with the protein sample, and the protein will interact with this lipid moiety, you can see some cases there is a rupture of the lipid, because those are the toxin molecule, they are interacting with the lipid and they are forming some kind of lipid protein cluster. Some cases you can see when that lipid chain length is increasing, so 10 PC means total lipid chain length is 10 carbon atom is there, and then 12 means 12 carbon atom, 14 means 14 carbon atom. So here the chain length is increasing. When chain lengths are increasing, that rupture of that lipid and the lipid protein aggregation tendency is low. Here you can see when we are increasing the lipid chain length, so proteins are clustering on top of that lipid. Here when we have a 18 PC or 16 PC or 8 PC, you can see proteins only clustered on top of that lipid. When that lipid chain length is smaller, there is a tendency of rupture of the lipid and forming a lipid protein aggregation. So from this particular study we can see this kind of changes, that there is maybe a lipid protein aggregation is happening when there is a shorter chain length of the lipid or lipid is not packed properly. Okay, in the next one, that okay, there is a couple of more example where you can see this circular thing is our protein molecules which is interacting with the lipid membrane, and here these are the lipid membrane which is decorated with the protein sample, and you can see different pattern in presence of different kind of protein sample. Here we are showing that there is one membrane protein, this is the lipid outer membrane, and these are the Toll-C molecules which is interacting with this lipid. So we'll just go through that. Okay, those are again very nice negative staining images. If you just consider the well distributed, those toxins molecules, you can see individual proteins and circular protein molecules, they are separated from each other, and all the details, even sometime from this negative stain you can count how many protomers are connected. So if you have a proper staining in this, you can see there is a total octameric shape is there. So there are many sample, even this one also you can count how many protomers are there. So if you can make a nice negative staining, even you can say how many protomers are interacting together and what kind of complex they are forming, what is the difference. In this particular case, here you can see we are identifying a complex, something is forming a 11 mer, that 11 protomers comes together and forming this kind of complex. Okay, so we'll just go quickly to see this one. Again, there is a SEC profile that we discussed that. Okay, so most of the time we have to use, in this particular case, author purified that protein using the SEC, and so author selected 3 different peaks, and from there the author calculated the overall molecular weight of the protein and also on the SDS page. From this SDS page you can see it is a very pure band, you can visualize, which is near the 50 kilodalton. From this author suggested that okay, so 50 kilodalton is monomer, and from other biochemical study author identified that near the peak 2 author is getting the hexameric or heptameric peak. From this negative staining author pinpointed that okay, so in this particular case author monitored the peak 1, peak 2 and peak 3 separately. When they visualize that peak 1, peak 2 and peak 3, they can see that peak 1 basically coming near the void volume region where there is a most of the time aggregation comes. In this particular case we can see there is a multiple assembly, that 2 hexamer or 2 heptamer or 3 heptamers are coming together. So here you can see 2 heptamer, here you can see 3 heptamers are coming together, and now the peak 2 basically giving a homogeneous oligomer, and then we can see the top view and side view, and then peak 3 basically we have a 3 peaks. If we just go back here and you can see that based on our SEC profile, this is basically giving the some kind of higher order oligomer. This is giving us an higher order oligomer. This peak is giving higher order oligomer, this peak is giving actual oligomer, and this is basically monomer, which is not forming any kind of oligomer. So if you just see that one, you can see this by negative staining also. Here you can see some higher order assembly, here you can see only oligomeric structure, and here you can see some kind of monomeric structure. This protein is elongated shape protein. this elongated protein, you can see there are multiple monomers all over this grid. So from this negative staining, when you do some biochemical test, that you perform SDS page, you perform native page, you performed ACC, and then you are performing all this negative staining study, from where you can isolate different types of population and you can actually visualize those oligomerization. So some cases it is forming a higher order oligomer, some cases it is just forming a hexamer, some cases it is just a monomer. So you can visualize, you can segregate them clearly, and not even that, what is the shape of the monomer. In the last slide, if you remember, it is some kind of elongated shape you can see, and when it is forming a oligomer, there is a large central hole also, you can see it in that. So this is the beauty of that negative staining. You can perform this kind of negative stain. I showed you different example how you can implement this negative staining for different types of protein and what kind of information you can extract, and this you can implement in your research, in your thesis or in your project also. This is all about the negative staining, and next class we will start discussing about the cryo-EM sample preparation. Thank you all for your attention.